Tung, Associate Professor of Biology, an evolutionary anthropologist and geneticist. Jenny is revealing links between social environmental factors and genomic variation and how these connections impact health, well-being, and longevity. For her transformational work, she received one of this year's MacArthur Fellowships, so-called Genius Grants. So she has been designated a Duke Genius. Please join me in welcoming Professor Jenny Tung.
It's a real pleasure to be here today. I was actually class of 2003, undergrad at Duke, and hopefully I have some, all right, great, good to see you. All right, so I'm gonna start my talk today, taking page from Jonathan, with some articles in the news. These are a series of New York Times pieces that have come out over the past, 5 years or so because of a regular interest in the news about what's collectively called the social determinants of health. That's the idea that our social status and our levels of social integration are actually some of the most powerful predictors identified to date of our well-being, certainly, but also of our disease susceptibility and even how long we live. So in fact, this last article on the bottom right there is an editorial in the Times from just the last few weeks. How good is the evidence that social environmental factors predict how well we do? There's quite a lot of evidence, in fact, and it goes back over 100 years or more. But I think it's thrown into particularly stark relief when we consider some of the very, very large data sets that people have been able to analyze quite recently. So this isn't work I was associated with, but I think it really drives home the issues that we're talking about. So what it shows is life expectancy at age 40 as a function of income at the same age. And it's really striking, not only because it shows a very, very clear pattern, right? There's a big difference in how long people at the very top of income percentiles and the very bottom of income percentiles, how well they do. But also because this is actually a 1.4 billion person year data set. It's a huge data set. And it may be of particular interest to you because probably quite a few of you are in this data set. Anybody who is in the United States, filed taxes, has a social security number, you can check these off as you go, and was over 40 anytime between the year 1999 and 2014 is actually right here. So you can take a little bit of a closer look if you want and see where you fell at age 40 and kind of make a decision about how you want to pursue the rest of the evening. Okay, so one of the things that's really striking here is that it's not just that we see differences in life expectancy when we get to the really bottom levels of the income percentile, the really bottom, the low levels of social and socioeconomic status in the United States. That's where we might think about material deprivation hitting, right? But we actually see years differences in expected lifespan for people who are making $100,000 and people who are making $200,000. And these kinds of observations are what led a social epidemiologist, John Castle, about 40, 50 years ago now, to make this argument, that there are certain sorts of environmental predictors of health and disease susceptibility that we're all really familiar with. These are the ones you see public service announcements about, right? They're about being tired, they're about having poor diet, and so on. What he proposed is that there's this whole other set of environmental factors that we need to think about. And that's the presence of other members of the same species, or more generally, certain aspects of the social environment. About 20 years later, a couple sociologists put it like this. They said social factors such as socioeconomic status and social support are fundamental causes of disease, right? So these kinds of arguments highlight the importance of the social environment, but unfortunately, they don't do a whole lot for biologists like me in terms of figuring out what our research agenda ought to then be, right? We're talking about effects that clearly physically affect our bodies, but here we're not talking about any kind of actual physiological mechanism that we can sink, our hooks into, right? We can't use the usual tools of animal models and experimentation and careful dissection in the lab to study this kind of thing. Fortunately, we are not the only social mammals on this planet. So what I'm showing you here are videos of the 4 animal species that my lab currently studies to try and answer some of those outstanding questions that still linger from the long-standing work that's been done in humans. So on the top left there are rhesus macaques, establishing positive, affiliative social relationships with one another and cross-generationally. On the top right, those are baboons in Kenya who are having a less friendly conversation about relative status. On the bottom left-hand side, those are meerkats in the Kalahari Desert of South Africa who are figuring out how to socially interact with each other through play. And on the bottom right-hand side are Damaraland mole rats, a highly cooperative species that lives together in colonies and depends on other individuals to help dig tunnels and maintain the system. In the interest of time, I'm just going to talk about the non-human primates here, the two monkey species on the top, and I'll give you some of the takeaways from our work. So one of the advantages in studying species like these are, unlike humans, they allow us to follow them around every day. So that's what's happening here. This is our field assistant, Kinya Waretere, in the Amboseli ecosystem of Kenya. And what he's doing is writing down pretty much everything these animals are doing. And this is something that's been ongoing work at our field site actually since 1971. So we're located right here at that dot in Kenya. And this is work that I do with another faculty member at Duke, Susan Alberts, Beth Archie, now faculty member at Notre Dame, but PhD 05 from Duke, and Jean Altman from Princeton University, who established this study system in 1971, and we're now spanning our 9th generation of baboons that she started following at that time. Okay. Many of our findings about the social determinants of health in baboons are summarized in some of the recent articles that have come out about our study system. In particular, there's this beautiful article with some gorgeous pictures in it in Scientific American. So if you're interested in learning more about this, I would refer you to this article. But in brief, I'll tell you some of the highlights of what we found. The first thing is that, like humans, more socially integrated baboons live longer lives. In fact, the difference is, again, on the span of years, and if we translate that into human years, the kind of way we do like dog years or 7 human years, baboon years, about 3 human years, we're talking about differences on the scale of a decade or so in human terms. And these are in animals where everybody's basically eating the same diet, where everybody has equivalent access to healthcare because nobody has healthcare, and nobody, even in the almost 50-year span of the study, smokes or drinks or uses illicit drugs, right? And we still see these same kinds of effects. In fact, what we specifically observe is that female relationships with other females are quite important, and those are usually their kin. But female baboon relationships with male baboons are equally and independently as important. And it's actually the high status females who form the closest relationships with males. So what we're learning from this picture is that social relationships, like tasty things to eat and good sleeping sites and access to mates, are a resource that these animals feel the need to compete over. We're also finding some interesting patterns with respect to social status in these animals. In particular, we find some patterns that echo those in humans, where individuals who are low social status show physiological signatures of higher stress. But we actually also see that in the highest-ranking male baboons, who have to constantly jockey for position and defend their status through physical competition. What that's beginning to show us is that these males seem to be following some sort of, live fast, die young strategy. And that highlights that how social status is first attained and then how it's maintained can be really important in understanding how these sort of social environmental factors translate into downstream outcomes. Males seem to suffer a little bit from it, females don't, because males have to physically compete for it. Females follow a more nepotistic, family-based rank inheritance structure. We've also had the opportunity to learn quite a bit from our social science colleagues, and they inspired us to do an analysis a few years ago that asked about the developmental roots of these kinds of differences. In particular, we asked about all of the bad things that can happen to a baby baboon. And surprisingly and strikingly, what we found is that those individuals who experienced no major sources of adversity, were not born in low-status families, were not born to socially isolated mothers, did not lose their mothers, did not have a lot of competition with other siblings, lived on average almost 10 years longer than those kids who were born and exposed to repeated insults, 3 or more. And remember, that's something like 25 to 30 years difference in lifespan if we translate it to humans. So together, this type of research is suggesting to us that the relationship between social factors and health, disease, and lifespan outcomes are not specific to humans. They're not some sort of weird product of the societies we live in today, but instead are deeply embedded in our social history. And that's really an opportunity to use comparisons with other social mammals to ask about when, why, and who is most likely to be affected. In fact, at last glance, there are at least 13 different species in which this kind of relationship between natural lifespan and social relationships has been described. So finally, I'm going to turn very quickly to one of the other systems that I spend a lot of time working on, and these are captive rhesus macaques. These animals are the reason why I'm fairly convinced at this point that there really are direct causal physical effects of differences in the social environment on our cells and on our bodies. Okay? This has been a really hard thing to pin down in humans because the way that you would classically do that kind of thing is to do an experiment, right? We'd take everybody in this room, we'd take everybody's income, and then we'd randomize it, right? And then you guys would go home and then live the next 10 or 20 years of your life, and we'd follow you and see what would happen. And you can see that this is probably something that's very unlikely to fly in human populations. And we can't actually do that in the wild baboons we study either, but we can do it in captive rhesus macaques. Okay? The way that works is that if we take adult females and we put them together into a new social group, individuals who are introduced early become the highest ranking individuals. And then as they progressively are added to the social group, they just kind of fit in the social hierarchy below the individuals who are already there. We can replicate that pattern over a lot of social groups, right? Replication is key to believing that an effect is really there. And then we can actually then watch them for a year or so and then take all the alphas and put them together and let them reestablish a new hierarchy, right? And watch them again. So that allows us to watch individuals who are high status when they were also low status, and individuals who are low status when they were also high status, and ask, well, what happens when what you're doing is manipulating social status without changing diet, without changing the groups you live in, and without changing anything about healthcare or health risk behaviors? So in brief, I'll just tell you that by combining studies of animal behavior with genomic and immunological techniques, we now know that social status is causally implicated in how our immune systems work, including in the sort of scenarios in which our immune system is most important, that is, when our cells are actually defending against bacteria or viral infections. Further, we recently showed that even when social status changes, a genomic signature of social history remains. Our cells circulating in our blood somehow still know whether they were high or low status a year or so back. So another way to think about this is that even the cells in your blood carry some baggage. Okay. So I just want to summarize and say that most of the stuff I use to contextualize why we're doing this research is work that's being done by social scientists. But work by our group and by others, I think, increasingly indicates that our social environment is biological. And that, again, gives us the ability to use the deep time lens of evolution and the powerful tools emerging from molecular biology and genetics and genomics to study these processes in the hopes of understanding who's going to be most affected and how that process might work. And again, thanks. Thanks for having me.
Well, thanks very much. Those are clean living baboons you were working with.
Absolutely.
I must be hanging out with the wrong baboons. I don't know. Again, you know the routine. We have mics running the audience. Questions for Professor Tung? We have one down in the front here.
Hi, my name is Robert Fuqua. I was just curious, from your studies, would you rather be a senior living in an adult community, let's say in Sarasota, or would you rather be surrounded by family in a big area such as Chicago?
Whew, that's personal, especially when you ask about specific places like Sarasota.
That's why we reverted to your studies.
So I have aging parents. They are living outside the suburbs of Atlanta, and I would love for them to come be closer to me and to my toddler. But they're physically in pretty good shape, and they have a fantastic social network, right? They have friends. They go out more often than I do. And I think that is an incredibly healthy thing, and that I would be pretty selfish, actually, to ask them to give that up. I mean, fundamentally, it depends on the type of social environment that's most rewarding to the individual in question, right? There actually have been some attempts to do experiments, like sort of assigning people buddies to be social with, and by and large, those experiments haven't turned out very well. It turns out that socially engineering who we're integrated with doesn't seem to replace, for whatever reason, the bonds that we form naturally. There's a lot of work on this being done now in the UK, where they have actually, starting in 2018, 2017, they instituted their first minister for loneliness, actually in response to the growing problem of social isolation in the elderly. So I think there's a lot of work on this to still be done.
One last one. Would you say that it would be the amount of endorphins that somebody's able to produce then? That would be the best community for them?
Well, I don't know about endorphins, because endorphins make me think of like my parents are going on motorcycle rides and stuff like that. They're giving them big rushes. You know, I think there's still a lot we don't know about how the cells in the periphery, like our immune cells in our blood, even know that they happen to be in the body of a socially integrated or a socially isolated individual. That's a big mystery, actually. But the best candidates so far are actually about hormones that actually regulate our stress levels, not about signals that regulate sort of our physiological highs. Like, so chronic stress is what we think is bad for you, as opposed to maybe a lack of endogenous opioids.
And have you seen connections? You know, there is research, for example, looking at the impact of pets, for example, on humans,
Yeah.
and some suggestion that, you know, when you have those kind of close relationships, the animals and humans both respond biologically in positive ways to those kinds of bonds. Is that something that fits within this framework that you're describing?
I think it could. You know, those are really fascinating sets of studies. They're hard to do in a really controlled fashion. You can imagine, like, if you're not a cat person and someone's like, I've randomized you into the cat treatment, right? That wouldn't actually work very well. And
Yeah,
so this
imagine
is
how the cats feel.
Yeah, exactly.
Yeah.
Well, right.
Yeah.
You're right. I should have thought about that too. Those sorts of the sort of studies that we want to do, the randomized control studies that say, in the absence, controlling for all these other factors, how well is this going to help you, are just really difficult to do in humans. So I think that there's interest in that area, and we'll probably, again, learn more as the studies get better and larger. But right now, I think it's unclear. Actually, I think one of the reasons to study the biological imprint of social relationships is because then we have this kind of somewhat objective measure of did it do anything?
Right.
Right.
Other questions? We have one right over here, middle of the row.
Thank you so much. That was so interesting. My name's IJ Eco. I was Trinity 2009. I don't know if you're familiar with the weathering hypothesis by Hieronymus. It's the theory that adverse effects for women, especially women of color, black women, increases their risk of, so that, I guess, the social structures they face as young women then leads to poor birth outcomes for the next generation. So premature, lower birth weight.
Yeah.
And I was wondering if you've seen correlations in the monkeys you've been studying, since it's more controlled compared to what we're able to see based off of just studying humans. I was just wondering.
We just published a paper a month or so ago on the intergenerational effects of early adversity in the baboons. So because they have shorter lifespans than humans, we can, you know, do this work in a little bit of a shorter amount of time. And what we found is that females who experienced a lot of early adversity grew up to be adults. So now we're talking like 10 years, 15 years later. Their environment has changed. They have babies themselves. Those babies are less likely to survive. What it looks like to us is that that is a consequence of their moms, the ones who actually experience that early adversity. And those babies are less likely to survive controlling for what they themselves experienced, to be clear. We think, although it's a question that's going to require, I don't know, another 50 years of research maybe, that this is probably because those females who experienced a lot of early adversity grew up to be in worse condition themselves, that they have these long-lasting effects from their development that actually magnify the effects of social adversity into the next generation. Yeah.
We have a question over here.
Hi, my name's Kay Hubbard, and I'm Trinity 08. So my question is about the Kaiser Permanente ACEs study,
Yep.
and if that informed any of your work.
Totally.
Also, I know that Dr. Joy DeGroux is a social worker who's done some work around epigenetics and trauma, and I just wondered if you see anything like that in your studies.
Yeah, so the ACEs study, which is a study of adverse childhood experiences in humans, is, for those of you who don't know, used this giant data set from Kaiser Permanente to show that major sources of adversity for human children predicted all kinds of negative life outcomes later in life. And that's actually exactly the model we had in mind when we did this, this study that I talked about in baboons, where we just said, okay, basically, how many of the baboon version of ACEs did you experience, and are we seeing parallel kinds of outcomes in the baboons? So it's one of the many ways that I really profited from the interdisciplinary environment at Duke, because I was raised as an evolutionary geneticist, and we don't read that kind of work. But in having conversations with my colleagues in sociology and economics and so on, they said, have you looked at this? And that motivated a whole area of research that we probably wouldn't have pursued, at least not in that particular way, without those kinds of conversations. In terms of epigenetic signals, yet something we're quite interested in. So my lab does a lot of work on the biology of gene regulation, and epigenetics is part of that process. And so we've been asking whether early life sources of adversity are detectable in the DNA of individuals who experienced it later on, and chemical marks on the DNA. I am not sure it is yet. What we're seeing is a lot of heterogeneity, where a lot of the social factors that we know that are most important to lifespan actually don't seem to leave at least a very long-term signal that we can pick up. But we do see some evidence that I'm reasonably convinced of that actually drought, so that's actually sort of, again, material deprivation does. So we're still exploring those sorts of questions.
We have time for one more question over on this side of the house. Yeah.
Hi, Danny O'Shaughnessy, Trinity 89. This is really interesting stuff, and thank you.
Thanks.
It made me curious if there are studies on twins in the U.S. where you have separated kids who have the same genome and have different adverse studies, if those have the same impact on people who are genetically similar or identical, and if that can be measured.
I don't know if that. Twin studies, which are used actually a lot in genetics, are really hard to get large sample sizes in. And twin studies with separation at birth, and here I think you're thinking about identical twin studies, which are even harder, are even more difficult. So I would have to ask my social science colleagues whether there's anything quite like that. There are a lot of adoption studies that have been influential in this area that don't use twins. Perhaps some of the most famous work was done on orphanages in Romania, where they see a lot of long-term negative health outcomes, and particularly mental health outcomes from sort of familial separation, and it's interpreted to be also lack of normal contact with adults. But again, not a twin study, so not exactly what you're asking for. So.
Well, please join me in thanking Professor Jenny Tung.